Hi, everyone. My name is Ernesto Sanchez. I'm with the Chemical Accounts Manager for Thermo Fisher Scientific here in Western Canada. Um, I'd been asked by the C45 Summit to introduce a panel of folks from, experts around Canada and to discuss current trends in lab data in the cannabis industry. Um, I've been in this role for 4 years, and like I said, just working with all of these experts on the panel. And I'll start with from out east. We've got Hubert Marceau. He's the Director of Development at Phytochemia.
Good day. So I'm Hubert, and I'm one of the founder and owner of Laboratory Phytochemia, and I work mainly in development, which include chemical development. And we've been working in the cannabis industry since 2014.
Awesome, thank you. Bridget.
Yeah, hello everybody. My name is Bridget Simons. I'm the founder and operating executive for Craft Labs, and we are essentially a QA consultants group that specialize in cannabis cultivation and processing startup. And we particularly specialize in laboratory test requisitions and using the data derived from labs to really home in on product specifications.
Awesome. Emily.
Hi, I'm Emily Kirkham with National Laboratory Services, and I'm president and founder of that company. We are consultants in the cannabis, but also many different industries in the laboratory world, such as environmental and pharma. I actually started in the cannabis industry back in 2014, and I started a company there that was also doing consulting, mainly for building laboratories for testing in the cannabis space. We were also trying to get a licensed lab at the time, but we had to transition to just only consulting through National Lab Services. But I've also worked in many other laboratory areas, such as government and environmental and pharma over the last 20 years.
Awesome, thank you. And Brian Shaw.
Hello, my name's Brian Shaw. I work with Caro Analytical Services. We're Western Canada focused. We have our head office in Richmond, BC, with locations in Burnaby, Kelowna, and Edmonton. We've been involved with the cannabis industry since the inception of the Health Canada regulations and legalization. And we're an analytical testing laboratory, so we do the full panel testing for all the different matrices, flower, oil, edibles, etc. And actually, Emily, who's on the panel, was super influential in developing our internal programs. So I'm sure we have a little bit of overlap there.
Awesome, thank you. And Erin Miller.
Everyone, I'm Erin Miller. I work for Analytical Labs. We are located in Vancouver, and we also have a lab in Mississauga, Ontario. I'm the QA director, and we test obviously cannabis and cannabis formulated products. Nandia was founded in 2013 and is now owned wholly by Aurora Cannabis. So we used to test samples for microprocessors, patients, but now we test exclusively for Aurora Cannabis.
Super, thank you. Yeah, and so I know, the discussion today is going to be based on, some of the current trends that we're seeing in lab data and things like that, but, also some of the issues and, experiences that we've all had in those testing areas. And I just wanted to, discuss with, the panel today. I know one of the issues that we're having currently in this industry is that there's no real set in stone methods, like There's ASTM and EPA type methods across the board for other applications, but cannabis is still relatively new, and because of that, there are pharma based implications, and the problem around that is that it's a plant-based issue as opposed to like a synthetically made product. So if any of you would like to kind of discuss kind of how or what you're seeing maybe towards how we're going to find a way to get methods being shared across the board.
Could I speak to that for a little bit, Ernesto?
Yes, absolutely.
So I think this is a very layered question, and you have to kind of look at it from the top down. And I think some of my other colleagues on the panel, maybe you can look at some of the more nuanced laboratory-based problems when it comes to consistency. But right now, our regulatory body is not requiring any level of accreditation across the labs. So whether that is ISO or GMP or however that's going to look, having that level it would really allow for some consistency across the labs, because what that does is it ensures that every lab is going to have a quality system in place and appropriate document controls. And that will go a long way, even before we have like a regulated certain method that we have to use for different products. And then you take one sort of further step into the layers, and you kind of need to look at the methods themselves. I mean, we're, like Ernesto said, we're all using different methodologies. And I think it would be great if we could have more sharing of these methods. They're not necessarily something we have to like really hold near and dear to our hearts. And I think if the regulators came in and said, look, here are some prescribed elements that you have to do in the method. These are the important things that you need to do. And then everything else is up to your own laboratory development. That would go a long way in the consistency of results. And maybe I'll let other people speak to more of the other levels of this. But again, yeah, we also need to look at this product as a plant, not a pharmaceutical product as well.
Yeah, I was just going to jump in and just say, I think speaking to what Emily's talking about is I'm hoping what can come out of this C45, maybe just starting from this panel. I think the Q&A will be really important when we get together in September, later in September, but is there a way to put together a working group who can sort of put forth potential methods, standardization of testing? To this point, I realize that there's often validation data is shared. And so that's sort of a way that they're trying to even the playing field. But to what Emily is speaking about is how do we value external accreditation? Should ISO and CALA labs, bodies that we're accredited under, should that trump the requirement for the validation? And I think you're putting a lot of onus on the producers to thoroughly check each lab. And that's, there's organizations who already do that. And I imagine a number of those companies would rather not have to take on that responsibility. And, those programs have items or policies like working with proficiency testing. And so that's kind of a nice way to even the playing field across all labs, is everyone's now validating their methods through an external sample. And that's a really nice way to be able to just bring everybody together into a standard approach and compare. And so I think that's something that we need to consider a little more thoroughly.
Yeah, I think that one of the biggest problem is the lack of a reference standard or standard material available to the lab, because I disagree that there's a lack of method, because since the mid-2000s, there have been analytical methods that were published and fully validated ones, such as the one from De Backer from Belgium University. And ASTM and AOAC have also been working on their methods. There is also the method from the German Pharmacopoeia that was published this year. I think that USB is also working on one. So the race the method race that is going on will make that there's a lot of method from different bodies on the market and in the literature also. But what's lacking is the way to confirm that when we apply these methods in the lab, we offer the right results. And the way to do this usually is to have certified reference material, which is still very scarce on the market, or ring testing in the case of lab also, which is currently also pretty uncommon, at least in Canada.
Yeah, because one of the things that that when I spoke to each of you prior to this too was around specifications for micro. How as the industry looking at that, and I got the impression that things are constantly changing around that. So is there something that you'd like to see in that space that could help you guys understand that portion of the analysis a little bit better too?
Yeah, I'd be interested to hear if other, oh, sorry, I was just going to say, I'm interested to hear if other groups on the panel have had this sort of shift in requirements. Seems to change over the last 2 years, let's say, where initially everyone was on the same page, and gradually, I don't know what the precipitous is. I believe it has to do with just maybe understanding the regulations or having more time to review. But the way it's sort of set up now is that it references, as we mentioned, the USP, the EP, et cetera. But there's lots of USP and EP monograms depending on what you're testing for. And so I've noticed that clients are starting to get more involved and deeper into those methods, and the standard they need to hit is sort of a moving target. And that's difficult from a laboratory perspective, because when you set up a method, you're sort of validating towards a certain number. And I'm just curious if the rest of the group has been struggling with this as well, and if they have any thoughts on maybe what's driving the change.
I think that one of the problematic regarding these limits is that in the regulation, they are worded in a way that the final decision on which of the monograph to use is left to the discretion of the transformator to based on what will be the end use of the product. And us as a lab, we don't always know what will be the end use of a product, because if you are from a grower that will sell its product and that will be sold and eventually packaged or extracted or everything, then you don't have the control over this chain of custody, because you are not part of it. You are kind of third party and external. So having all these monographs with all these parameters available to us is very hard to have a good idea on what to test for. And I think that most producers also are a bit fuzzy on this, because they don't always know exactly what will be the end product also. So what we hear a lot is that especially in micro, is basically use the monograph that will allow me to pass. And I'm not sure if this is the right way to work for. And the other thing to consider is that the monographs are not cannabis specific, and therefore may not be adapted to the plant itself, which may have some peculiarity that other vegetable material don't have, for example.
I'd like to add a perspective from the LP. I represent a few LPs right now. Product specification is an absolute requirement ahead of sending samples to labs, because if you don't know what you are testing, to what measure to then sell it into the supply chain, you will fail on deriving meaning from the lab information, the lab result tests. However, the industry over the last 2 years, Brian mentioned the changes in it, and here are the big changes that have influenced how specifications are derived on products. One is that the growers grow, and then after they harvest and start testing, then they choose often what subchain the product goes into, not the start of cultivation trying to hit a particular product category and a specification. It's the reverse. Secondly, there is major influences in the industry based on the considerations around irradiation and the considerations around proper moisture content of products. So those are kind of the equal forces that are coming back on why microbe analysis and or other lab testing is this fuzzy moving mark. I think when it comes down to it, there are no mature supply chains yet. By maturity, I mean like rugged history. You're putting the same into the same supply chain over history, where the LP can then derive, here is my QC, high, median, and low, and that has not had enough history behind it to know that. So you're fighting that particular battle over the next, startup to 2 to 3 years.
I know both Erin and Emily wanted to add to that too. So why don't we start with Erin?
Yeah, I mean, I don't really have anything new to add except to say we've seen the exact same thing in Anandia, that there is a big struggle with choosing what specs to test to, especially with micro, for the same reasons that Hubert and Brian mentioned, that there is no cannabis monograph. That is something we would like to take on through the C45 lab committee to make a monograph specific to cannabis and to help make it clear and related to the use. So the LPs might be making a product that's going to be used in many ways, maybe for a vape and so what specs do they test to, and trying to piece together different pharmacopoeias that are not cannabis specific, it's very challenging.
Yeah, so I agree with what everybody else is saying here on the panel. I'm really happy to have somebody like Bridget who you're working with the LPs, because I couldn't agree more with what you're saying about how the LPs have to know what they're testing for, but quite often they're looking to the labs for that answer, and the labs are really not equipped to provide that information. We don't know what they're going to be using the product for. And again, I would say, looking back at the regulators, and maybe we can, from this group of the C45, we can try to promote this idea of looking at this for maybe a cannabis-specific monograph. We can look to other jurisdictions that are more mature, like California, that have been around for a long time, and they've really taken a more realistic approach to micro-testing within the product.
I know Bridget mentioned something there about lab costs and how they don't always apply these until the end. And this was something that you had mentioned to me as well, Emily. So if you guys wanted to talk about that, and a lot of these companies and clients are saying, "Oh, well, these lab costs, is it really that expensive?" And if you guys want to elaborate on that and why these processes might be more costly than people are saying, they don't get to see the hidden costs all the time. So if you'd like to elaborate on that.
So yeah, I mean, we get a question about this quite often, and in my group, we've been building cannabis labs since 2014. So we have a pretty good understanding of what it costs to put all this together, and that's something that as Bridget spoke to, like looking at the cost that analytical testing adds to this product, and there is a lot of infrastructure behind what goes into a lab and getting it properly set up. And sometimes I think there was a lot of people jumping on the bandwagon saying, "Oh, this is going to be a big moneymaker getting into labs," but they don't really understand what has to go into it when you're talking about $250,000 for instruments, and all these trained staff that have to have degrees, all the quality systems that have to be in place, and then on top of which the validation process, which developing and validating a method, especially for something that is very complicated like pesticides in a complicated matrix like cannabis. I mean, it's a 6-month process even for experienced analysts. So when you look at these costs, and if you don't have different revenue streams in the lab, that can be very detrimental to the lab. And yet, there's always going to be price driving down. We've seen that in other jurisdictions like Colorado. So having some consistency and requiring, again, sort of a uniform accreditation would really sort of go a long way to having like a level playing field. One thing I really would like to see in this industry, and it might be a little bit pie in the sky, but there is this pay per THC, which really, we've seen it in many other jurisdictions where people are always searching for the highest THC number because they get the most value out of their product, whether it's being put into extraction or just being sold. And it really causes a problem in the industry because, the producers are, and we understand the tight, situation that they're under. The higher the THC, the more value they're going to get out of their product. But, we can't have, groups searching around for the highest THC number from different labs. So this is a problem. And I'm not quite sure beyond like establishing something like the wheat board or like our, maple syrup situation in Quebec, like how do you get around something like this? So maybe I'm looking to this panel, maybe you have some ideas about, how do we get rid of this problem?
Well, I do have an idea, but it's also quite blunt, is to get rid of the THC level. It makes no sense to report on a package for significant number on the THC level. In my very own opinion, there should be a range like 0 THC, 0 to 5 THC, 5 to 10, etc., 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 which would be a lot more representative of what the consumer is buying or the processor is buying, because just due to the inherent variation inside the lot, added to the inherent variation of the sampling, to the inherent variation of the method, it doesn't make sense to report 4 significant digits on a product.
Two parts to this. I think a lot of what we've already talked about will go along with it. Accreditation and reference materials, proficiency tests. That'll all help the lab. But I think the second part of it is going to be an education. And this is more difficult and probably going to take a lot longer, but to make people understand that essentially the higher THC is not really what they're after anyways. I think for so long people thought they were buying a high THC product, but I think we've all probably experienced it. Label claim is inflated compared to the actual THC content. And so experience is really all relative. And I think people are thinking they're having an experience with a certain level of THC, but it's actually a lot lower THC. So I think this is going to take a lot of time because this is ingrained in people. But I think that's a huge part of it. It's this misconception that higher THC products are better.
Come on, people, just smoke some more.
I think there's a double-edged sword here also to consider, is that one of the reasons why they are looking for high THC is because this is what sells. Speaking with people who are more on the retail sale, they would tell me that if you give the choice between a 21% and a 21.12%, the customer will automatically go to the 21.12% without asking any question in a lot of places. So we are in a very immature market where the customer is used to another way of consumption in a lot of jurisdiction, where you need a little to get more. And I do believe that the fact that there are no publicities that can be done on cannabis is actually a little bit detrimental in this effect, in the sense that you cannot educate the consumer to consume more smartly, if I can say that, if you cannot explain what is the product. However, the trade-off is that you don't really want more consumer because it's still kind of an artificial paradise. Just like you don't want more alcohol consumer on the market, you don't really want more cannabis consumer in the market, but you want them to consume better. So we are kind of at the beginning of this new market, a new industry, where nobody has really a good idea on where it will go. And the consumer is just clinging to this whole idea that I need to have less for more. And this is what drives these. I only want the highest possible THC content and disregard everything else.
Yeah, and I think, and someone on the panel can probably help me here. My understanding is that it's essentially one test per batch, and the batch size can vary greatly depending on the size of the producer. And is that the case? Because to me, to try to nail down, like Hubert said, a 4-digit significant figures on your THC doesn't make any sense when you're looking at potentially what would be in the tons of flour. And is that how it works?
The mature LPs will have a proper sampling and batch plan. And I think that's, condition here in the industry is to help customers and LPs build proper sampling and lot size characterization.
Sorry, the EP has a sampling protocol for vegetal material that is usually one that we suggest. However, they don't test for homogeneity. I know that some jurisdiction in the US, among other, will require the producer to test for homogeneity in their batch, basically sending at least 2 batch to the lab, and then if the 2 batches are too different, then doing retake. But I believe, and I'm pretty sure that in Canada, this is not the case at the moment.
No, you have to set specifications, median, high, low, which when I mean by high, low, it's what's your tolerance of standard deviation around the spec. And that only comes with history and growing your cultivar in your space. It doesn't come by writing a protocol and trying to hit ask the SOP, which is what leaders are surveying presently. It comes by growing history of the cultivars, not in a room with a bunch of cultivars, but growing history, designing what the median spec is, designing what you're going to allow tolerance above and below, and then informing the lab that this is in spec, these ranges are out of spec for THC, CPA, CBD, CBDA. Moisture is another huge topic of confusion. And we're talking about terpenes yet, but the thought is that the more terpenes I have, the better my product is. And I think everybody here will agree that the qualitative versus quantitative analysis of terpenes to the consumer's knowledge of what terpenes mean to them is a very large gap still we need to close.
I just to speak to this idea of like the batch size again, I couldn't agree more with what you guys are saying. And this is also an area where I actually agree with the regulation approach to it. And a lot of producers have talked to me and asked like, why hasn't the regulator specified batch size or a sampling protocol? And it really is like a risk-based analysis that has to be done at the producer level. Like Bridget was saying, you have to look at what you're growing and how you're sampling. And if you are sampling a huge lot and only testing once, there's a high risk involved in that. So, there's a financial incentive on that side. It may cost you more to test more within the batch or more often, more frequently. But, if you have a failed result from a very large sample, that is a huge risk of that. So that I think has to be really done at the producer level, and that's some part I think we need to agree with.
Yeah, well, what I was talking about when I say managing unknown versus eliminating unknown is that there are a lot of QA in this industry who's coming from a pharmaceutical background, and there are a lot of companies who consider cannabis to be a pharmaceutical, but a plant is not a pharmaceutical, and you cannot manage it the same way that you do for a pills, for example, where you know exactly what are the ingredients that come into the composition, and if you see something unknown, then there's a problem into the process. When we're talking about a plant, there are a lot more unknown than there are known. So this is a little bit what Bridget was saying, going with history, is that this is where statistics get very useful. If you just look at the acceptable variation on the active ingredient, for example, if you look into a pill, you want to have less than 5% on each side. If you look at the plant, USB has published some general recommendation in February, and they're suggesting plus or minus 20% on a lot. If you look at the last proficiency test that was done by Acala, the 1 sigma deviation was 8%, which means that if you go to 2 sigma, then you have plus or minus 16% of each side. So you cannot expect a plant to have the same consistency as a pharmaceutical, and this is something that needs to be taken into account when evaluating the value of the final product also. To give you an idea, plus or minus 20% on a 15% THC product means that the product can be between 12 and 80%. So that's a very large range if you consider that the threshold are at 1%, are very strict at -1 or 2%. So this is where I think that there is an education to be done, that you need to manage unknowns. You need to do statistical testing on your product. You need to do ANOVA to be able to see what are the impact of each parameter, to really be able to conscript your product to a set of parameter, and as Bridget said, your mid, your low, and your high, and from there act upon the outside, the out of spec value, more than the small variation that may be surprising for you but are normal when you're talking about a plant product.
Yeah, and I think if you don't mind, I just jump in here. You know, when it comes to the question of how do I know that I'm doing the right thing or that my results are valid, and you know, we can draw some information from other industries. Food industry, for example, is subjected to random testing, so off the shelf. Those samples will come in and be tested to confirm that it meets regulations for pesticides, metals, other contaminants. And similarly, in the environmental industry, there's an organization, even just through BC alone, who is the BC Laboratory Advisory Group. And when there's new methodologies, it's always discussed in those groups, and there's interlaboratory testing that goes on. And so I think one thing that hopefully can come out of this conversation in this group is that we could set something up similarly where we are willing to share a sample between labs, and we all test and compare results. And what would that have allowed to do is both confirm that we're doing what we should be doing, but also give us a chance to discuss maybe differences that in our methods that might contribute to any change or variation that you see in that result. And that would help develop more robust methods. And that's something that we could all certainly communicate on afterwards.
If I may, there is something that I would like to share with all labs here, is the cannabis industry it's quite special in the sense that it's kind of the merging of multiple industries together. We're talking about the pharmaceutical on one side, the agronomical industry on the other side, the natural product industry on the other side. And there's an industry that is often overlooked and that probably has a bit to bring on the table also, and it's the cosmetic industry. And the reason for that is, especially in the high-end level of the cosmetic industry, they are used to work with plants that they know they will change every year, and they are used to manage these variations. such as the one that we can find in cannabis. So I think that being able to open our horizon to other industry and the way that each of them work or approach their problematics is probably what will grow the most the cannabis industry in the world.
Okay, well, thank you everyone for joining the C45 Summit panel to discuss some, current trends in the lab space with us here. Hopefully, our audience will have some great questions for us at a later date. And once again, thank you for everyone's time.
Thank you. Have a nice afternoon.
Bye.
Bye.
Thank you.
Great. Thanks, guys.